Okay, so I will just go straight to this diagram because the lesser gene takes place in this part. You have to understand what is going on before we talk about the lesser gene. So the main purpose of doing genetic engineering is that we want to put human DNA into the bacteria so that the bacteria can produce some of the proteins that we want. But because bacteria does not have the DNA or the gene to give instruction for that type of protein synthesis, we want the DNA from the human host cell. So what happened is like we do some stats, and the last stat is that we want the recombinant DNA. All right, we want this stat, and then we are going to put this recombinant DNA into the bacteria. All right, so first what we need is the vector. Vector is the DNA molecule of the bacteria that can accept a foreign DNA. So this is a vector. You can also say bacteria plasmid. All right, so this plasmid will accept the foreign DNA. So what is a foreign DNA? A foreign DNA is the DNA from the host cell. So DNA from human body is called a foreign DNA for the vector, and then we are going to combine these two. But before we don't go to combining these genes, we will go to this. So as you see, before we get a recombinant DNA, what we need to do is we need to cut the vector and also complementary DNA. Restriction enzyme will cut the vector and also complementary DNA. So here in this step, when restriction enzyme cut the vector, what happened is this. So vector is also a DNA molecule. It's not exactly DNA, but it fashion like DNA, and it also has similar structure like DNA. So literally, vector is also made out of nucleotide. Okay, so instead of thinking on nucleotide, let's say that DNA molecule is a gene. So let's say this is a DNA molecule. It is a gene. We have a gene here. Throughout the gene, we have different types of genes here. The genes that can control various types of functions. But here, what we are going to talk about is the LATZ gene. So on the bacteria vector, at some point we have a LATZ gene. And now I just say that the restriction enzyme is going to cut the vector, right? So where did the vector cut the what did the restriction enzyme cut the vector? The restriction enzyme cut the vector at the LATZ gene. So what happened is that after restriction enzyme cut the vector, the LATZ gene is broken. Okay, so you have to remember this one. It is broken already. From this stat, LATZ gene is broken already. So we do the other stats. We got the recombinant DNA. Now we put the recombinant DNA into the bacteria, and we are going to let the bacteria multiply because we want the bacteria with recombinant DNA to multiply, right? We want a lot of bacteria with recombinant DNA. But the thing is, when we do let the bacteria multiply, not all the bacteria has the recombinant DNA. Only some of them has the recombinant DNA. So how do we identify which one has recombinant DNA and which one does not have recombinant DNA? So bacteria with rDNA, bacteria without rDNA, how do we identify this? So with recombinant DNA will give a white color, and without recombinant DNA will give the blue color. That is because the one without recombinant DNA, it has the intact LATZ gene, which means that those bacteria without recombinant DNA, they has a vector, and that vector has not been broken by the has not been broken by the restriction enzyme. Do you get what I mean? In the beginning, the restriction enzyme will cut the vector at LATZ gene, so the LATZ gene will be broken. LATZ gene being broken means that this vector is going to combine with the complementary DNA from the host cells, and it's going to form the recombinant DNA. So any bacteria that has recombinant DNA has the broken LATZ gene. All right. So for this bacteria with recombinant DNA, Less gene is already broken. So less gene broken, it gives a white color solution. But less gene intact, it gives a blue color. All right, not solution, just white and blue. So let's read this. I will show you. So this one, the bacteria that contain plasmid with intact lysogen. Plasmid with intact lysogen means that lysogen is not being broken down by the restriction enzyme. It means because restriction enzyme didn't cut the plasmid only has the plasmid and no complementary DNA. So it's not going to form the recombinant DNA. All right, so this is a chemical reaction. So they produce the enzyme beta-galactosidase, and this hydrolyzes the A scale. A scale is what we need when we do the screening. So because lysogen is not broken, it can synthesize the enzyme beta-galactosidase, and this galactosidase react with the A scale that we use in the screening, and it gives the blue color. All right. And then the bacteria which contain recombinant DNA. It means restriction enzyme has already broken down the vector, and the vector has already combined with the complementary DNA and formed the recombinant DNA. So any bacteria that has recombinant DNA has the broken lysogen. And because the lysogen is already broken, it cannot give instructions to synthesize this enzyme. So literally, bacteria with recombinant DNA do not produce this enzyme beta-galactosidase. So beta-galactosidase cannot react with A scale. So as a result, bacteria with recombinant DNA cannot form the blue color. Instead, it only form the white color. All right.